Good day, everyone. I am Joster Pakibo, and today I'm going to report about viral evolution, horizontal gene transfer. Let's start by defining horizontal gene transfer. Horizontal gene transfer is an exchange of genetic material between existing cells in the same generation. And horizontal gene transfer, or HGT, has three methods in transferring genetic material, and these are conjugation, transformation, and transduction. And among these three methods, we will only discuss what is transduction, since we are talking about viral evolution, and among these three, only transduction deals with viruses. So what is transduction? Transduction is a process by which two bacteria share genetic material between themselves in the same generation by a virus or a viral vector. And among the three methods of transferring genetic material, transduction is the only method that does not require physical contact between the cell donating DNA or the donor cell and the cell receiving DNA or the recipient cell. So to fully understand what is transduction, here, these two are bacterial cells. And this yellow color bacterial cell is what we call the donor cell, and the other one is what we call the recipient cell. So the genetic material from the donor cell will transfer to the recipient cell by the help of viral vectors, or what we call bacteriophage. There are two types of transduction. First is the generalized transduction, and next is a specialized transduction. Generalized transduction is a process of transferring genetic material that is considered to be pure bacterial DNA from one cell, which is the donor cell, to another cell, or the recipient cell. And this is carried out by virulent bacteriophage. And while specialized transduction, this process transfer not pure bacterial DNA, and this is carried out by temperate bacteriophages. So to fully understand what happens during generalized transduction, it starts with the bacterial cell that we call the donor cell. And we all know that bacterial cell contains its own genome. And in this case, it contains two genes, the A plus gene and the B plus gene. So this means that this bacterial cell here expresses these two genes properly and produce products related to A plus and B plus. Now, here at the top of the cell is a viral vector we called bacteriophage, which infects the cell by injecting its own genetic content inside. After injecting, inside the cell, phage enzymes are produced that breaks down the host DNA into smaller fragments. After this, the phage DNA particles will start to replicate, and then products will be produced such as protein coats. During the formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. Let's take a look to all of these assembled phages inside the bacterial cell. One phage head has the A plus gene and the other has the B plus gene. And the rest, they just have the phage DNA. Then after the maturation of these phages, it will then be released from the bacterial cell. And this bacterial cell will undergo lysis, leaving the cell dead open. Now, all of these phages carrying this DNA will now infect other bacterial cells. In this case, a phage containing an A plus gene infects this another bacterial cell containing a negative gene. Now, this phage with this A plus gene incorporates its genetic material to the bacterial cell genome, and by recombination, this bacterial cell will have this A plus gene and will produce products related to A plus gene. And that's how generalized transduction happens. An example of bacteriophage that mediates generalized transduction is the phage T4. Phage T4 infects Escherichia coli bacteria, and this has a double-stranded DNA, and they came from the subfamily Tevenverinae from the family Myoveridae. And phage T4 is capable of undergoing only a lytic life cycle. That's why this type of phage is a virulent virus. This is a summary what happens during generalized transduction. First, a virulent bacteriophage infects a bacterium. Then, the phage genome enters the bacterial cell. Next, virus governs the bacterial metabolic mechanisms to make its own DNA and other necessary components and enzymes. And later on, bacterial DNA hydrolyzes into small pieces. Then genetic material packs inside the new phages. Occasionally, bacterial DNA fragments packed in new phage capsids. Then bacterial cell lyses and releases the new phages. And lastly, when transduced phage infects another bacterium, the previous bacterial DNA incorporates into a new. So let's move on to specialized transduction as to how this type of transduction happens. In specialized transduction, it also started with the same mechanism as the generalized transduction, where we have this bacterial cell containing bacterial DNA. In this example, we can say that this bacterial cell has been successfully infected by a bacteriophage because of the presence of prophage, as you can see. This prophage is actually a viral genetic material incorporated into the genome of the bacteria, and this prophage able to produce phages if it is specifically activated. This cell with prophage may remain in this state for several generations without releasing it from the bacterial genome. You may ask why this bacterial cell still survived when it was infected successfully. Well, this is because the bacterial vectors in specialized transduction are only temperate phages. Temperate phages is the ability of bacteriophage to display lysogenic life cycle, which means they can integrate their genome into their target cell without killing it, which also means that there is a stable integration of phage genome into the bacterial genome. So when this phage, which has lysogenic cycle, is exposed to a stimuli such as some chemical or UV lights, it causes induction of virus genome from the host cell. And this begins the lytic cycle. Lytic cycle, in order to release the phages, the donor or the host cell will undergo destruction of the cell membrane and eventually kill the donor cell. Lytic cycle is somewhat opposite of lysogenic cycle. Now, upon the induction of prophage, on a very rare occasion, a piece of bacterial DNA near the specific site of insertion remains attached to the phage DNA, and a piece of phage DNA is left behind. And then the replication will follow, and then the assembly of all phages will take place inside the bacterial cell. And then finally, the phages will be released from the bacterial cell by cell lysis. Because of this very rare accident or error, these phages are defective. They are considered defective because they do not carry the entire phage genome, but still they can infect other cells, and that is by their attachment and injecting their nucleic acid into the bacterial cell. Now both phage and bacterial DNA become integrated into the new host chromosomes. Now, only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction. An example of temperate virus that mediates specialized transduction is the phage lambda, or it is officially called as Escherichia virus lambda. This virus infects Escherichia coli bacteria, and it has double-stranded linear DNA. And since this is a temperate virus, it is capable of undergoing lytic and lysogenic life cycle. And according to studies, lambda phage injects their nucleic acid for at most 15 seconds. And since its DNA are linear, and a linear DNA is susceptible in digestion and degradation of the host cell, after it injects its DNA into the bacterial host, the DNA, the viral DNA or the DNA of the phage lambda circularizes. And how this happened? The two cohesive ends base pair are ligated together by a host enzyme DNA ligase, forming a circle, which makes this connection permanent. During this point, the lambda phage will start to go under prophage, and then the lysogenic and lytic life cycle goes on. To summarize specialized transduction, first, a temperate bacteriophage infects a bacterium. Then viral DNA integrates into bacterial chromosome and becomes the prophage stage. Then viral DNA remains within bacteria for several generations. Upon a spontaneous induction, viral DNA detaches the bacterial chromosomal DNA. Next, fragments of bacterial DNA detach from the bacterial chromosome with viral DNA. Next, viral DNA replicates together with bacterial genes and package inside a new capsids and make new phages. Then bacterial cell lyses and releases the new phages. Then phages infect new bacteria. And lastly, bacterial DNA mixes with new bacteria during infection. This table shows the differences between generalized and specialized transduction. First, we know that generalized is carried out by virulent bacteriophages, and while specialized is done by temperate phages. And in their life cycle, because of virulent bacteriophages, generalized transduction undergoes lytic cycle, and in specialized, it undergoes lysogenic cycle. Well, in lysis of bacteria, bacterial cells in generalized lysis quickly, while in specialized, bacterial cells are not lysed quickly. It even survives for several generations. And in packaging of genetic material, a portion of the donor bacterial DNA is enclosed within viral capsid in generalized transduction, while in specialized, small parts of bacterial DNA remains attached to viral DNA during the detachment from the bacterial chromosome and are packed into new capsids. In integration of viral DNA, the viral DNA in generalized is not integrated into the bacterial chromosome, while in specialized, Bacterial and viral DNA are both integrates. And then in hydrolysis of bacterial DNA hydrolyzes into pieces by virus in the generalized, while in specialized, bacterial DNA do not hydrolyze. And then the production of prophage, we know that there is no prophage formation in generalized. And then in specialized, prophages are formed during the very first of the infection. Though I have discussed what is lytic cycle and lysogenic cycle, this photo summarizes the differences of these two life cycle of bacteriophages. So what is the role of transduction in antibiotic resistance? Antibiotic therapy represents one of the most important medical advances of the 20th century, and it is a very valuable resource in combating infectious diseases. But somehow, this antibiotic against infectious disease became ineffective, and this is because of the emergence of antibiotic-resistant bacteria. There are many naturally occurring ways in which susceptible bacteria became resistant to antibiotics, and that includes horizontal gene transfer, which is the transduction. The acquired gene from the donor cell carried out by a phage, this could be from the phage DNA or the bacterial DNA, may be a useful gene for the bacterial host to produce specific protein against antibiotic, and eventually the bacterial cells survive. These newly acquired genes may prevent the antibiotic from entering the host cell, pump antibiotic out from the cell, or they destroy this antibiotic by an enzymatic reaction inside the cell, or even modified the antibiotic's target so that it won't no longer binds with the antibiotic. Once a certain bacterial cell acquire these characteristics from the transferred gene, now they could be resistant to the antibiotic. And since horizontal gene transfer is continuously happening, this antibiotic-resistant gene will be passed from one bacterial cell to another, making the antibiotic ineffective in some ways. And this is thought to be the major means by which bacteria evolve and acquire antibiotic resistance and virulence factors that accelerate the emergence of new and progressively more pathogenic strains. These are the references of this report. And that is all for viral evolution horizontal gene transfer. Thank you for listening and have a great day.